it's really a pleasure for me to introduce Ting. She's a friend as well as the previous speakers. We've been working together for maybe 3 years now, have exchanged people in the labs, so we have a lot of connections. But one of the first things that struck me most when I first met Ting was obviously her kindness. She's a very kind person. But at the same time, her way of thinking about science, which is always out of the box type of thing. She always goes for the strange things rather than the mainstream things. And that shows on her CV. She's been director of a few programs, or some programs over the years, including, and I have to read it, the Consortium for Space Genetics, which strikes me as quite interesting. At the same time, she is a dear leader and probably the founder, one of the founders of the Personal Genetics Education Program, which tries to educate the general public on what genetics and genomics can do for their lives in many aspects, going from telling the Hollywood guys how to do the movies correctly to going to church and explain the people who believe in something that science is also there, probably. Many things in this genetics program, education program that she runs. She's very well known for oligo painting, these little pieces of oligos that helps us to visualize the genome. And I think she's going to be telling more about this in her 30 minutes talk, including how fortunate we were, and I include myself into this list, of getting funded by the NIH through the Center of Excellence for Genomic Sciences. Her center is called the Center for Genome Imaging and has been funded, a month ago, Ting? So we started only a month ago, officially speaking, but we have done a lot of work, and I think Ting can tell us a bit more about this work. So Ting, thank you for being here, and the floor is yours.
Mark, thank you so much for your friendship, first of all. It was a transformative event when I met you at Woods Hole. And thank you for mentioning our space genetics program. We are actually focusing on climate change now, and for mentioning PGED. It's been, I'd be happy to answer questions about those too. But really, thank you, Mark, for such a generous introduction. It's quite a privilege to be part of this program. So thank you for the invitation. And I just want to say that it's thrilled that you're in the audience, because this is the first time that I will be publicly acknowledging our Center for Genome Imaging, which Mark and I share with Nicole Linaretti and Erez Lieberman Aiden. And as Mark said, we started just a few weeks ago. Now, our center is part of a number of this program called Centers of Excellence in Genomic Science, which is funded by the NHGRI, the National Human Genome Research Institute. The demands of being in the center is you have to have extreme innovation. We got rejected the first time round. You need to focus on critical issues in science and or medicine. They highly encourage multi-investigator interdisciplinary efforts, and they put a serious focus on education and outreach. There's not much more infrastructure to talk about, except they want you to innovate and then to disseminate. So here is a list of the centers that are currently in existence. The program's been around for a while, so you can look at their website to see the other ones. And I'll tell you a little bit more about what the center has meant for us. Now, I'll move now to the science and come back to this just a little bit later on. What is our mission? The mission of the center that I share with 3 wonderful PIs is to develop, implement, and disseminate imaging, analysis, and modeling tools for elucidating the 3D organization of genomes in their entirety. So why entirety? Because the genome is an integrated unit. It replicates as a unit, it goes into mitosis as a unit, and it segregates as a unit. And actually, it was observing the entire genome that enabled Fleming, Bovary, and Sutton to figure out that the genome is involved in heredity, thereby helping to launch modern genetics. Now, the genome also integrates its function. So at the maternal to zygotic transition, hundreds to thousands of genes are simultaneously activated in embryogenesis across many, many species in the living kingdom. Well, there are, we could spend a long time talking about the countless networks that are coordinated by the genome-wide. The one I want to focus on today, though, is ultra-conservation. Ultra-conserved, it's a mystery. Ultra-conserved elements were discovered about a decade ago by, 2 decades ago by Gil Bejarano when he was in David Hauser's group. He set off to look for genes that are sequences that are over 200 bases long and are 100% identical between human, mouse, and rat, fully expecting that nothing would come out of this search because our knowledge of mutation rates, fixation rates, and drift prohibited such sequences from existing, but he found 481. And then shortly thereafter, Sandelin et al. And Wolf et al. Confirmed the existence of these really surprising sequences. They encompass regulatory functions, protein functions, a number of functions, but none of these can explain ultra-conservation, either alone or together. And after many studies, including a very recent tour de force by Snetkova et al. From the Wiesel, Panacchio, and Dickel groups, studying the potential of these ultra-conserved elements to be ultra-conserved because they are regulating important genes, the conclusion is that actually gene regulation can't explain why some of these sequences have remained essentially unchanged for 300 to 500 million years. So it's a mystery. Now, they are located all over the genome. And so we need to come up with a mechanism, perhaps, that is genome-wide. And our model in the lab is that allelic UCEs pair up. They're quite unique. They pair up with each other. And when they pair up, they compare themselves to each other. And if a point mutation is found, or deletion or duplication, or there's an inversion or translocation that prevents these alleles from pairing up, then you have the cell or the organism induces loss of fitness, which removes these changes from the genome. And this would ultimately drive ultra-conservation. So it's a model. We're still not sure we're right. We're always prepared to discover that we're wrong. But right now, we're in the middle of testing 896 human UCEs to see whether they follow the prediction, which is to avoid being deleted and duplicated. Now, what Adnan Durti, Charles Chang, Ruth McCall, Yelena Ertzeg, and most recently Tae Ryu, in collaboration with Claire Redding and Harrison Brand and Mike Talkowski, what they've been doing is looking at the countless copy number variation data sets that are now known for generally healthy individuals and asking, are UCEs in those deletions and duplications or not? And what we have found is an extraordinary avoidance of UCEs from falling into copy number variants. What we do is we take our copy number variants from a data set, and then we ask, what is the overlap between those and UCEs? Then we compare that overlap to the distribution of overlaps that are obtained when we do 1,000 random throws of sequences that are matched to UCEs in length and number. And then we compare the distribution to what we actually obtain. Now, what Tay has done recently is to look at the NOMAD data set. And here is what she got. This is the distribution of overlaps or the random throws. And here is the real overlap we get with those copy number variants. As you can see, they're extremely depleted. So what does this mean? This means that if you took a reference genome and you threw onto it all the copy number variants from any number of people that you want in your data set, there would be gaps. And we think it's in these gaps that UCs tend to reside. This model predicts that if you do have breaks or deletions near UCEs that disrupt their pairing, then you will find that those rearrangements are associated with disease. We've done a number of studies. This is perhaps the most dramatic. This is a study of 118 trios that fall on the autism spectrum and their unaffected parents. This is a data set from Volto von Silfhout. Because we have the trios, we can parse the copy number variants in the affected individuals into those that they inherited from their unaffected parents and those that are de novo in their own genomes. And what we saw was pretty compelling. The inherited CNVs were quite depleted of UCEs, but the de novo CNVs were actually enriched in UCEs. We have also looked at balance rearrangements, inversions and translocations that don't necessarily delete or duplicate anything from the genome. sort of conceptually imagining that if they were to pair up these chromosomes, there would be buccal points. And so what we asked is, what's the distribution of UCEs relative to these buccal points that may prevent pairing? And we got the most extreme enrichment. Not surprising, because perhaps by our model, because these rearrangements come from a data set representing individuals with neural developmental disorders, a whole range, including many severe developmental disorders. Now, at the time, oh, I have one more thing to say about this. Ruth then asked if she looked at different size flanks in which she was looking for these UCEs, what is the relationship of UCEs to those breakpoints? And it turns out the closer you got to the breakpoint, the smaller the flank that is, the more enriched the UCEs were. Now, at that time, we didn't have a controlled data set of translocation and inversion breakpoints associated with healthy individuals. But since then, those data sets have become available. And this is where Tay has been working closely with Harrison and Mike. And here are her results. So the orange bars of what you've seen already. And the green and the blue represent the breakpoints of inversions and translocations and the enrichment for UCEs. Remember, these are from healthy individuals. And close to the breakpoint, she essentially saw no deviation, no significant deviation from expected. It's only when she got quite distant to it that she started to see some enrichment. And that's a little puzzle we're working on now. You may ask yourself what happens when we divide the NDD original data set into inversions and translocations. And what you see is the enrichment close to the breakpoint is maintained. So we're pretty excited about what's happening here. So to summarize very briefly, we think this may be telling us that ultra-conserved elements, even if they have other functions, such as for regulating genes, that they may be like centromeres and telomeres, and that their job is to protect the integrity of the genome. And they do that by pairing and comparing themselves allelically. All right, so does pairing really happen in mammalian cells? There was a lot of doubt about that many years ago, but right now there is quite a good list of pretty impressive phenomena, all of which have been implicated in some form of pairing. So I think that pairing is something that happens in mammalian cells. Now, we study pairing in Drosophila because in Drosophila, throughout most of development, the somatic cells pair their homologs end to end. And here I'll tell you about a story that's been spearheaded by Jumana Al-Hajabed and Yelena Erceg, who's now at UConn, in close collaboration with Anton Gold-Barroco, who's at the IMBA, and we collaborated with him when he was in Leonid's group, and we also worked with Brian Lejeune when he was in Job Decker's group. Now, what Yelena and Jumana did was to conduct haplotype resolved Hi-C, meaning that when they looked at the interactions between genomic segments, they were able to characterize those interactions with regard to the parental origin of the interacting fragments. So here's a Hi-C map with the maternal paternal chromosomes separated. You see the dominant Hi-C dominant diagonal that you see in Hi-C generally, but you also see these off-diagonal diagonals that represent interhomolog interactions. And when we looked closely, binning and measuring the pairing, it looked like there were two kinds of pairing, at least, tight pairing and loose pairing, which correspond to two different kinds of Hi-C structures. So here's a type region that is tightly paired. And do you see this really dark diagonal? We think that that diagonal represents a railroad track pairing between the homologs, very close and very in pretty good register. But now look at the loose pairing. You don't see that diagonal at all. This has only been seen, this had only been seen once before by Egan when he was looking at polytene nuclei. And we think what this means is that the homologs are near each other, so you can pick up some interactions, but they're not really aligned that closely. So they're kind of nearby, but not too close. All right, can we see this? So we have in the lab for a long time realized that genetics, as powerful as it is, which has a resolution of one base pair, only gets you so far in understanding the three-dimensional organization of chromosomes. So we've been developing imaging technologies, and our technologies really rest on a really rich history of innovation, a lot inspired by Rob Singer's group, attaching oligos to fluorophores straight through to Yamada and all, and Boyle et al.'s use of synthesized oligos. Here I'll tell you about work that Brian Bellevone with Eric Joyce pioneered in the lab with early contributions from Ben Williams. Brian's now at UWash and Eric's at UPenn, and Ben is at the Helmsley Charitable Trust. Now, oligo paints are computationally designed oligos with a region of genomic homology flanked on one or both sides by non-genomic sequences onto which we can park a variety of functions. And one of the things that Brian did was to collaborate with Xiaowei Zhuang's group, in particular Alistair Bedecker, and with Peng Yin's group, in particular Ralph and Meyer, to develop two forms of super resolution, which we call oligostorm and oligodNA paint. In addition, we've benefited from Stephen Wang's work and Xiaowei Zhuang's group for using these streets to affect sequential hybridization. All right, so Alistair's now at Stanford. Ralph has moved on, oh my goodness, to Vienna, no, to the Max Planck, and Meyer is at a company called Inveio. Okay, so oligostorm, talk about that a little bit, is a single molecule localization microscopy developed by Xiaowei Zhuang, in where single blinks of fluorophores are recorded singly. And you collect these blinks into an XYZ table, and then you computationally form these XYZ coordinates into a three-dimensional structure, as you see here. This is actually a 290 kb loop with 10 kb anchors and a 270 kb body. After looking at a variety of genomic reasons, we assembled quite a team. This is a wonderful team. He brought on Irene Farabella from Marc's group, Cynthia Perez-Estrada from Erez's group. We've worked very closely with Bruker, so there's Carl Ebeling and Jeff Stuckey, and Dean Lee from my own lab to really take advantage of this technology. And as we looked at loops and compartments and TADs, we only wanted to image more and more of the genome. And it became very clear that we wanted, in order to understand the genome, which functions as an integrated unit, we'd have to see the whole thing. But the technology as it was back then would have required 63 days to image a single nucleus, which is not very good and not very affordable. And so we've been busy developing technologies. And I think right now we're on the threshold, perhaps, of being able to image many nuclei in 3 to 5 days. This would not have been possible, though, without forming this consortium. Officially, as of a month ago, but I would say over the past few years. Now, to put things into context, about a decade ago, this was the best microscope I had in my lab, a dissection scope, and here was the most powerful and highly powerful computation that we did. I had a very heavily Drosophila genetics lab. Today, we do single molecule localization, microscopes more complicated, and intense computational analysis involving image processing, modeling, AI, machine learning, two different kinds of modeling. And here are the Marc, Erez, and Nicola, with whom we have really truly benefited. And I want to point out that this consortium, working together with these groups, has truly deepened our knowledge. It's diversified our experiments. It's accelerated our science. And I think everyone in my group will say that it's truly enriched our personal journeys. So I really want to thank my consortium partners. All right. So oligostorm can walk along megabases of DNA. Here are the two homologs of chromosome 19, about 8.16 megabases. The thing about imaging is it gives you real measures of distance, surface area, sphericity, volume, overlap, density, and the context within the larger nucleus. What we've done is to take those parameters and mix it with the exquisite high-C results. This is, I really want to acknowledge Erez and Cynthia here. And what Irene did is to put these two together under the umbrella of her imager program. And by doing that, she, was able to model the entire 8.16 megabase region. And our hope ultimately is to do this for the entire genome. All right, so now let's go back to pairing briefly. Here's Jumana. She is looking at various regions of the Drosophila genome, those that are tightly paired, those that are not so tightly paired. And what she's asking is there a common feature that defines tightly paired regions and loosely paired regions. Now, we Drosophilus have been trying to see pairing for over 100 years. Most recently, Catoni et al., Zaba et al., Mateo et al. Have been using higher resolution fish, very high resolution fish, and I'll tell you what we've been doing with Oligostorm. Jumana has compiled hundreds of images. This is a region that I've split into 4 segments. The middle 2 segments are tightly paired, the outer 2 are loosely paired. And instead of seeing a side-by-side arrangement for loosely paired and railroad track for tightly paired, we saw essentially equal entanglement for both types of pairing. So we think what this means is maybe loosely paired regions are not really side-by-side. They are entangled, just not in great register. We thought about this a little bit more. We thought maybe railroad track pairing stiffens the chromosome fibers. So is there something else that imaging can tell us about the structure of paired regions? And in fact, there is. It turns out paired regions have a higher radius of gyration. This is what we're trying now to integrate into the relationship between tightly paired regions and both lowly expressed and highly expressed regions. And the interesting fact that lowly expressed regions are loosely paired regions are only lowly expressed. And might this, I guess we're asking now, is might this rigidity or radius of gyration explain this kind of segregation of structure with gene expression? We're also looking at another kind of pairing, that between sister chromatids. And here I want to acknowledge work by Nuno Martens and his collaborator Tatsuo Fukugawa. They are looking at the chicken centromeres Z and 5 because these 2 centromeres are composed only of unique sequences in the chicken. I'm going to really skip ahead and show you some images of the left arm of centromere, just outside of centromere 5, this tiny region here. If I walked you through it step by step, you would see that there are 2 sister chromatids here. The cartoon on the right shows you the 2 sister chromatids and the regions where the sisters touch. Here's the right arm on the other side of centromere 5. The sisters are a little more separated here, and here's the cartoon showing how those sisters touch. And here is the whole region put together. Now, what Nuno's done is looked at many, many of these regions. He selected out, in this case, just those nuclei in mitosis and asked a question that came out the opposite of what we anticipated. He took each region and he said, what's the percent of that region that sits right in the middle of the centromere core? And to our surprise, the centromere core is the one that is least in the centromere core. It's the flanks that are in the centromere core. And his interpretation is this, that when the cells are poised to go in mitosis, the centromere cores are separated and the flanks actually zigzag back and forth between them. So Nuno's busy looking at this, and we're really hoping to integrate Hi-C, hopefully with Rez's help and Tatsuo's help, and bring an imager to see if we can better understand centromere structure. Okay. So as you can imagine, our desire to see more and more regions only increases with time. What is the challenge? So here is the challenge. And I think it's going to be our challenge for the next few years. Maybe by the end of our KEGS funding, we will have made some progress. So Oligostorm. Oligostorm, as I explained before, is the compilation of single blinks. So you capture a region, you get a bunch of blinks, then you computationally put those blinks into a structure. Then you move on to the next section. You collect a bunch more blinks one by one. And then after you've collected those blinks, well, then you computationally put them into a structure. Then you move on to the next step, and so on and so forth, collecting blinks one at a time, and then assembling them into a structure, which I have to say is just exquisite. They're exquisitely beautiful to look at. The resolution's fantastic, gives you volumetric information, gives you entanglement information at high resolution. Extremely, extremely informative structure, but slow. On the other hand, you can actually image very quickly if you let all the fluorophores blink. You get one big bright spot, you get a single centroid from that spot. Then you move to the next step. All these single fluorophores are on at the same time. You get another centroid, and then you move on to the next step, get one big blink, and you get a centroid, and then you connect those centroids. We call this method the ball and stick method. Very fast. You can emit hundreds of thousands of cells in a very small amount of time. Data storage costs are not that great. And I really want to acknowledge work done by Wang et al., Bintu and Mateo et al., and Cardozo, Ghisi et al., which have really established two methods called ORCA and HIAM. For the whole genome, we're thinking we need to combine, well, maybe one strategy is to combine fast, ball and stick with volumetric, and then image, use this combination to get a whole genome super resolution image. We then import imager to model to a very fine genomic resolution. So step one is to have to make ball and stick genome wide. Currently it's used locally, but we need to have it be genome wide. And I'm very excited to say that a year ago, Hoi Wen, Shantanu Chattaraj, and David Castillo in Marc's lab collaborated together to demonstrate the capacity of a technique they call oligophasique to image genome wide. And then I also want to acknowledge very exciting papers from Xiaowei Zhuang's lab and Long Kai's lab, Fei Chen, Jason Ben Ostro, and Ed Boyden's group, paper by Payne, Chang, and Paul Reganato. These papers demonstrate the capacity to look at between 1,000 and over 3,000 targets at the same time. I'll just talk about oligophasique for now. So oligophasique is the fusion of oligopaints and a technique called phasique. What happened is that Sunny, who was a graduate student in my lab at the time, was talking with Evan Darty, who was a graduate student with George Church, and they realized that the method that Evan was developing, phasique, to look at RNAs could be combined if we put the barcodes that were being used to identify the sequencing, that was being used to identify the RNAs, have that sequencing sequence barcodes on the oligopaints. We're very excited about this. Then Sunny graduated. He actually joined Eric at UPenn, and Evan started a company. But at that time, thank goodness, Hoi and Shantanu joined the lab, and when they got together with David, things really started to move. The advantage of sequencing is that contrary to sequential hybridization, where the total number of targets you can look at is the product of, of the number of fluorophores and the number of rounds of querying, oligophasique, the total number of targets you can look at is F to the power of N. So you can get into the thousands pretty quickly. 7 rounds of sequencing, you get 16,000 targets. I won't go through the details of it, but here's a cell tipped over on its side with the barcodes of sequencing above it. Then you decode the barcodes, and I say it casually, it is a lot of work. This is partly why the CGI is so important. The computational piece, merging with the imaging piece has been essential for us. Here was what we published, 129 targets and their localization. We had a little barcode issue here, so we couldn't disambiguate perfectly, but we demonstrated genome-wide pretty clearly. This is 1,000 targets that Antonio's, reflecting a library that Antonio's Liotas has generated. It's 1,000 targets, each target being about 50 kb in size. And we are now working on this piece. I'm going to show you one image. Hoi and Shantanu have generated this. This is the, to the right are the sequencing rounds, which are overlaid here to the left. And right now, David, Hoi, and Shantanu are working very hard on really, hopefully, a very effective method for quickly decoding these barcodes. All right, so that's the goal. Take ball and stick, such as oligophasique, and then attach that to super resolution, which will give us volumetric information. Does that work? In fact, it does. Here is a super resolution image looking at 66 spots, which all look alike. And at this point, at our super resolution, we had no idea which spot belonged to which chromosome or which target. So what Hoi, Shantanu, and David did was to then take that image, take that slide, and run it through just 4 rounds of oligophasique, which by decoding the barcodes, unambiguously identified each one of those spots to a target. So whereas we would had to image for days to get each one of these spots one at a time, this was done in a matter of, well, this decoding was done in, the super resolution was done in 2 hours as versus days of super resolution. And here are the kind of tables that we're hoping to generate. These are all the targets from that region. I think part of the cell might have gone off the edge of the slide, but we believe that now this will be a computational playground that should keep us busy for years. I'll finish up by saying that the next step is to go for the whole genome. And, you know, some days we think we're there. Other days we realize we have to be a lot smarter. I have a team that just never gives up. Again, here's Antonios. He designed a library. Sarah has brought in a level of imaging acumen that I cannot even begin to describe. And Faye joined us recently. She brings in engineering. Here's a library that they're working on. I will show you short walks along the ends of chromosome 1 and on chromosome 21. The point of this library actually was to know how to space the walks and how many steps we can take. So here are the P and Q arm spots of chromosome 1. They're separate because they're at the other ends of the chromosome. Here's a contiguous walk of several megabases along chromosome 21. As you can see, there's going to be a lot of data here. The resolution is fantastic. I want to just end by saying we are extremely interested in volumetric information, surface information, entanglement information. Are there holes in the middle of this? Are they hollow? Are they solid? And so that's the place where we are at. And I think I should be ending. So I'll end now. I cannot tell you how lucky I feel every day to be working with the people that I passed through my lab and who are currently there. Their level of persistence in the face of, I have them here in front of Niagara Falls, in the face of technical challenges is really admirable. I've mentioned most of the key people here by name already. Here's my current lab to the left. I haven't yet mentioned Connor. Connor is just joined the lab early this year. He is interested in moving our technology into tissues, and he's also developing technology. Arjun Patel joined this week. He is going to be working in the technology development area. And I just wanted to say Mohammed Hanan is the one who keeps our lab running all the time. Sometimes we kid ourselves, we say we co-parent. I think Mohammed sometimes single parents, especially during this pandemic. I cannot say enough about our collaborators. I mentioned today our collaboration with Mike Talkowski, Leonid, Yob, George, Peng, Xiaowei, and Tatsuo Fukugawa. Most recently, we started to collaborate more closely with Genmon, not only in the 4DN, but outside the 4DN, and with Harris Lewin and his team. And then, of course, it's the Marc, Erez, and Nicolas group with whom I really have the honor of running this CGI. Finally, many thanks to our IT community at Harvard Medical School. Super resolution imaging generates truckloads of data. We have been innovating with IT on how to handle those data. Happy to talk to people about that. We're funded by NIH through a variety of sources. This is the first time I've been able to put kegs on there. I want to say that I am a non-equity holding co-founder of Acuity, for which Hoy and Shamtanu are going to serve as CSO and CTO starting in December. Lots of acknowledgements to Bruker that has supported us in kind and also with a sponsored research agreement. And earlier on, I worked closely with Recor and with MicroArray. And with that, I think I'll close, and I'm happy to answer any questions about the centers of excellence, how to get them, what they're really looking for. I can tell you, Mark and I can tell you a lot about how we failed the first time round. So if you go for one of these, you don't have to go through what we went through, or anything about pairing or imaging. Thank you very much.
The first question we have from Ilya Flammer. Is it possible that ultra-conserved elements are involved in meiotic chromosome pairing? And that's why you get such a big difference between inherited and de novo CNVs.
We are so excited about that possibility. So Tay, I'll start the imaging side. Tay is now designing libraries with Antonios that should, and with Hoy and Shamtanu, that should allow us to look at where those ultra-conserved elements are in 3D in various kinds of cell types. we think meiosis is a perfect place. In fact, we think of meiosis, I know we think about meiosis as generating egg and sperm, but in our lab, we also think of meiosis as one of those random tests of population diversity. A random pick male and a random pick female. You put the genomes together, it gives you an assay of what kind of diversity is in the population. And we think about meiosis as a place where that particular cell may be able to adjust diversity based on that, perhaps by demise, perhaps by surviving. We don't know. So it's a long answer to your question, but yes, I think meiosis is very interesting.
A very small question from me. Is it known something about the relationship of ultra-conserved elements and CTCF binding sites?
So we are looking at a variety of epigenetic sites. And you know what? I should know that answer. Tay, if you're on, if you could write the answer into the chat box.
Yes, so I have tried to search CTCF binding sites within UC sequences, total like 1 kb around UC sequences, but I didn't find any significant enrichment.
As he's bringing Daniel up, I just want to say that UCEs are one of the longest standing mysteries. This is other people's words, had to come out of the genome era. And we like our model, we're testing it, but truly the real answer may come out from a completely unexpected place. It's a fun place for thinking.
Hi, Ting. Great talk and great data. Another question on ultra-conserved element. Do you find this type of element in Drosophila? First, I mean, of course, it's what you show is in mammals, but did you find similar sequence in Drosophila, and could that be involved in somatic pairing, or is it something totally absurd?
No, we love pairing. So here's the thing. Since ultra-conserved elements are computationally defined, you can, as long as you change that definition, you can find them. A number of studies have been done in Drosophila where the demand for conservation can be lessened by shortening the sequence or allowing not having to have perfect conservation. So you can find them.
Okay.
Do they behave the same way as in mammals? Not strikingly so, but I think this is an area that still has to be explored. And I have to say that I'm embarrassed to say I have not kept up deeply with it. I would say they've looked in plants also, just to expand your question a bit. And I think the divergence time was much greater than between human, mouse, and rat. They lessened, again, the identity requirement, and they found a few dozen ultra-conserved elements that interestingly were depleted from copy number variants in plants. So it is kind of a circular argument. You define it, then you study. But I'll just throw out one last thing. We are wondering if ultra-conservation didn't just suddenly appear, let's say, at the junction of birds, reptiles, and mammals, but that it's co-opted other functions. We know in organisms, there's a repair mechanism that does depend on looking at strands. And so it could be that various versions of ultra-conservation have embedded themselves down different branches of evolution. And so we will see different manifestations of it. Right now, Taya and I are really enjoying establishing collaboration with Jen Ma's group. Jen, I haven't talked about what we've been doing with Yang, but you know what we're doing, and also with Harrison Lewin's group. I think there's a lot of place here for really interesting computational work, and our lab is very open to collaborating with anyone in this area. So just write us. And if I don't answer right away, it's because I'm very bad at email. Just keep writing. And
maybe just a comment regarding your imaging of somatic pairing in Drosophila. So as you know, I mean, we are also working on the modeling the pairing, and actually it's beautiful because I mean that's something we never really look at carefully. But I think if we look at our simulation of pairing and tight pairing and loose pairing, we actually do see that tight pairing is increasing the effective rigidity of the chromatin. And so
great.
that's beautiful that you can see this in the data.
Daniel, I didn't know it was you. I just knew it was a Daniel. I actually,
Yeah, it's me.
I was just talking with Jumana about contacting you. So let's talk. I would love to.
Yeah, that's
to do more
something
about
we can discuss this.
that would be great. Thank you for that information.